Moving to the next question. Kran's anatomy can be seen in Kran's anatomy. In the plant anatomy chapter, anatomy of flowering plants. There is a special type of anatomy that is known as Kran's anatomy that is seen in some monocot plants, not all the monocot plants, some monocot plants. It is known as Kran's anatomy. What is meant by Kran's anatomy? Kran's anatomy is a wreath-like arrangement of bundle sheath cell. Generally, the vascular bundle of leaves, they are conjoint, collateral, closed with end dark xylem. The vascular bundle is conjoint, collateral, closed with end dark xylem. Then the characteristics of leaf is the vascular bundle is surrounded by a bundle sheath. That means a peculiar type of cells which surrounded with a vascular bundle, and they are known as bundle sheath. So there is a definite bundle sheath. Generally, these bundle sheath cells are devoid of chloroplast. Generally, they are devoid of chloroplast. But in the case of some plants, there shows Kran's anatomy. There is the bundle sheath cells are large, thick-walled, and they contain chloroplast also. So they are large and thick-walled and they contain chloroplast. And these large cells are arranged surrounded to the vascular bundle. They form bundle sheath. And because of this nature, they appear as a wreath-like structure. And the presence of these bundle sheath cells, which helps in a mode of nutrition or pathway, that is Hatch and Slack pathway. It increases the efficiency of the plant, efficiency of carbon dioxide fixation, that means the production of glucose. It increases the carbon dioxide fixation by means of a special type of anatomy that is known as Kran's anatomy, that is seen in plants. They are growing in equator region or high temperature tropical region. Some of the plants, they are growing in tropical regions and they are coming under a monocot plant group. There shows a special anatomy that is known as Kran's anatomy. Among these, potato, tomato, pea, these three are dicot plants, and there is only one plant, they are coming under monocot, that is a maize plant. So maize, sugarcane like plants, they shows Kran's anatomy, and these plants, they are highly or twice efficient than these C3 plants. Normal plants, that is known as C3 plants. Here the plant with the Kran's anatomy is known as C4 plants. Here the first stable compound is a 4-carbon compound, hence it is known as C4. In generally C3 plants, the first stable compound is a 3-carbon compound, hence it is known as C3 plants. So C4 plants, they are twice efficient than C3 plants because of the pathway that is known as Hatch and Slack pathway. This Hatch and Slack pathway is due to the Kran's anatomical features. So the answer is maize. Option 1. Clear? Then next, moving to the next question. Binomial nomenclature is introduced by. Actually, binomial nomenclature. Actually, the Carolus Linnaeus or Carl Linnaeus is generally known as father of taxonomy. It is because of his main contribution. One of the important contribution of Linnaeus is binomial nomenclature. What is binomial nomenclature? The term itself, it means a scientific name which contain two words. A scientific name which contain two words. The first word is genus name, and the second word is species name. That means there are some rules and regulations of giving a binomial nomenclature. So the main or the first rule is the binomial nomenclature, binomial name which contain two words. The first word is genus and the second word is a species name. Then the genus name written in capital or start with a capital, not written, it start with a capital letter, and the species name start with a small letter. Genus name start with a capital letter, species name start with a small letter. That means our scientific name in binomial nomenclature is Homo sapiens, the name Homo, which is genus name. So it start with the capital letter. Then species, that is sapiens, it start with a small letter. So genus name and species name, and it start with the capital and the small letter respectively. That is the first rules of binomial nomenclature proposed by Carl Linnaeus. Then next rules and regulation of binomial nomenclature is, then if the scientific name is printed, if the name is printed, it should be in italics. It is very easy to recognize from other words. If it is handwritten, it should be underlined separately. If the name is handwritten, it should be underlined separately. So, if the name is printed in publishing books, it should be in italics. If the name is handwritten, it should be underlined separately. That is the next rules of binomial nomenclature. Then the name should be Latinized. We can give the scientific name in different language or any language, but it should be Latinized. That means India Latinized to Indica. So Mangifera India. That is Latinized. When it is Latinized, it is converted to Mangifera Indica. So Indica is the Latinized version of India. So the name should be Latinized. Then next one is, there is an author citation. What is author citation? Author citation is the abbreviation of the name of the scientist who gave the scientific name to a particular plant or particular organism, that is written or printed in abbreviated form after binomial nomenclature or scientific nomenclature. That means after binomial nomenclature, there is an author citation which is started with a capital letter. So if the binomial nomenclature which was given by Linnaeus, there is an abbreviation of Linnaeus, that is LIN, which is start with the capital L. So the binomial nomenclature which was introduced by Carl Linnaeus, because of that he is considered as the father of taxonomy. So the answer is Carl Linnaeus, option 2. Okay, then moving to the next question. In trees growing in temperate region shows clear demarcation between spring wood and autumn wood. This is because spring wood and autumn wood. Spring wood and autumn wood. What are the difference between spring wood and autumn wood? The name itself given, that is spring wood or early wood, that is formed during spring season. Definitely. Similarly, autumn wood is formed during the autumn season or winter season, late autumn season or winter season. The wood which is formed late autumn season is known as autumn wood. The wood formed during the spring season is known as spring wood. Then what are the characteristics? Why these type of woods are formed? That means during spring season, cambium is highly active. Seasonal activities of cambium. That means during spring season, cambium is highly active. During the autumn season or winter season, cambium is less active. So, the activity of the cambium, which affected by the outer seasonal variation. Then, when the cambium is highly active, they produce a large number of secondary xylem having vessels with a large cavity. The vessels with the large cavities are formed during the spring season, that is known as spring wood. During the winter season or late autumn season, cambium is less active and they produce limited number of secondary xylem having vessels with a narrow cavity. So during winter season there is a narrow cavity wood is formed. Then during spring season there is a large cavity wood is formed. So these type of woods are called spring wood and autumn wood. So our option is the water stress is more. It is because of spring wood and autumn wood is formed. It is a wrong answer. The climatic conditions are uniform throughout the year. When the climatic condition is uniform, if there is no change in activity of cambium, there is no spring wood and autumn wood or different type of woods are not formed. So it is wrong. The climatic conditions are not uniform throughout the year. It is because of spring wood and autumn wood are formed. So the answer is option 3. Option 3 is the right answer. Then moving to the next question. In cloning vector, antibiotic resistant genes which are helpful. We heard about antibiotic resistant genes and also cloning vectors. In our second year chapter, biotechnology, principles and process, there is a diagrammatic representation of a cloning vector, that is PBR322. PBR322 is a cloning vector having two antibiotic resistant genes. One is ampicillin and one is tetracycline. There is a two region, tetracycline resistivity region and ampicillin resistivity region. These two antigen and two antibiotic resistivity genes, which helps to the selection of recombinant from non-recombinant. Selection of recombinant from non-recombinants. Selection of recombinant from non-recombinant, it is by the help of these antibiotic resistant genes. Transfer of foreign genes into the host by means of microinjection, then shotgun method, etc. Cleaving of factor by restriction endonuclease enzyme, then making the host cell competency, creating the competency, it is due to some activities. And it is not the effect of antibiotic resistant genes of a cloning vector? So the answer is selection of recombinants. In a cloning vector, antibiotic resistant genes which are helping in the selection of recombinant. Then, okay, clear. Moving to the next question. During chemiosmotic synthesis of ATP in photosynthesis. In photosynthesis, chemiosmotic synthesis of ATP or chemiosmotic hypothesis. Chemiosmotic hypothesis, it was proposed by Peter Mitchell. Peter Mitchell proposed a chemiosmotic hypothesis. What is meant by chemiosmotic hypothesis? Chemiosmotic hypothesis represented or proposed by Peter Mitchell. It demonstrates the formation of proton gradient within and outer the membrane, where it occurs, where it takes place. First, we understand that the structure of chloroplast. Structure of chloroplast. Chloroplast, we know the chloroplast is a double membrane bound cell organelle. Double membrane bound cell organelle. Chloroplast is a double membrane bound cell organelle. That means it has an outer membrane. So if this one is a chloroplast, it has an outer membrane. This one is an outer membrane. And there is also an inner membrane. So this is an inner membrane. These outer and inner membranes are quite different. Outer membrane is more permeable than inner membrane. Inner membrane is selectively permeable membrane. And there is a space in between outer and the inner membrane is known as periplastidal space. What is? Periplastidal space. The space in between outer and the inner membrane is known as periplastidal space. And there is a fluid filled internal space, or the space within the inner membrane is filled with a fluid, and the place is known as stroma. The stroma is a site of dark reaction, so there is many enzymes are present for this dark reaction. So this stroma region, that is the internal space of the chloroplast, or within the membrane there is a space, a fluid, that is known as fluid filled space is known as stroma. Within the stroma, many flattened sac-like structure which runs through the stroma of chloroplast. Many flattened sac-like structure which runs through the stroma is known as thylakoids. So these are thylakoids. Thylakoids are of two types: stroma thylakoid and grana thylakoids. What is stroma thylakoid and what is grana thylakoid? The thylakoids which are used for the formation of grana is known as, so the stacks of thylakoid is known as grana. So the thylakoid which is used for the formation of grana is known as grana thylakoid, and the two adjacent grana is interconnected with a thylakoid which is known as stroma thylakoids. Grana thylakoid and stroma thylakoid, they are of two different thylakoids. Then here the thylakoid is, this is a thylakoid, and the thylakoid has a membrane that is known as thylakoid membrane, and there is a space within the membrane that is known as a lumen, and the thylakoid membrane contain different photosystem 1 and photosystem 2. This thylakoid membrane contain different electron carriers or electron transporters. When an electron that is emitted from the water due to the splitting of water, this electron transported to the electron carrier and finally enter into the NAD, which is converted into NADH2. So this movement of electron which allow the trans allow the not only the transfer of electron and also it allow the movement of H plus ion across the thylakoid membrane. It increases the H plus concentration within the lumen. So this movement which helps in the increasing the hydrogen ion concentration within the lumen, that create a concentration gradient or proton gradient, that create a proton gradient. Actually, chemiosmotic hypothesis which explain how this proton gradient takes place. There are three conditions. First one is splitting of atom. Second one is conversion of NAD, which is reduces to NADH2. And the last one is an electron carrier which acts as not only an electron carrier, it acts as a proton carrier. It transfer or it passes H plus ion from the stroma region to thylakoid lumen. So thylakoid lumen having high H plus concentration. Stroma region, that means outside the thylakoid membrane, there is stroma region, there is less H plus concentration. So there is a tendency to move from H plus ion having a tendency to move from higher concentration to lower concentration. So this is facilitated only through the ATPase enzyme that is present in the thylakoid membrane. So this ATPase allow the movement of H plus ion from the thylakoid lumen to the stroma. So during this conversion, during the transportation of H plus to H plus ion, there is ADP plus inorganic phosphate converted to form ATP. That means ATP synthesis takes place within the stroma region. So this is explained by chemiosmotic hypothesis by Peter Michel. So the question is, during chemiosmotic synthesis of ATP in photosynthesis, the proton gradient is not required. For the synthesis of ATP, there is a proton gradient is necessary. The proton accumulate in the intermediate space of the chloroplast. No. Proton accumulation that takes place within the lumen of the thylakoids. Option 3. The proton accumulate within the lumen of thylakoid. And the proton accumulate in the intermediate membrane space of the mitochondria. Chemiosmotic hypothesis explain the ATP synthesis during the photosynthesis, not in respiration. So there is no mitochondria, there is a chloroplast which acts as the electron carrier and the electron transportation and also ATP synthesis. So here the answer is option 3, the proton accumulate within the lumen of the thylakoid. Okay, answer is 3. Okay, clear? So it's all about biology, especially in botany.